This is the B5 Triple Science Booster Pack. This is suitable to watch after you've watched the B5 in a nutshell, which is the double content. This boosts you up, giving you all the knowledge you need for separate sciences, triple science biology. Okay, first thing we're going to look at is the brain. Now, the course content tells us that we have to be able to identify the main parts of the brain on a diagram. We've got the cerebral cortex, okay, this large area at the top. Think of it as a large area because it's got lots to do. It's responsible for consciousness, intelligence, memory and language. We've got this area at the back, looks a bit like a lettuce leaf, the cerebellum. That coordinates movement and balance. We've got this section here, the medulla oblongata. It's long, oblongata, okay? And that's feeding down into the spinal cord, where we know we've got the central nervous system. This is our automatic actions, heartbeat and breathing. Out of interest on this diagram as well, we've also got the hypothalamus. That's responsible for temperature control, the thermoregulatory centre, and we talk about that when we talk about vasoconstriction and vasodilation. We've also got the pituitary gland, okay? This is the one that makes all of the hormones that begin with letters, FSH, LH, TSH, ADH, okay? They all come from the pituitary gland. At no point in time are we going to answer a question with the word brain. We must be specific. So neuroscientists map regions of the brain. It's really hard to study the brain because it's so complicated and it's so delicate. So if a part of the brain has been damaged, it can tell us a lot about what that part of the brain does. The brain can be stimulated using tiny electrodes. This will be done while a person is awake so that they can see the response that the person has to that stimulus. MRI scans are also able to give us detailed pictures of the brain structures. We're able to then compare those pictures we take with pictures that are either damaged or healthy, so we can make those comparisons. Moving on to the eye. Again, course content tells us that we have to identify some specific parts of the eye on a picture. So we've got this outer surface, which is really tough. If you saw the eye dissection, you'll know that it's really difficult to cut into. That's called the sclera. The front section of that, covering the lens and the pupil there, is the cornea. That part is able to refract light, so if we can change the shape of it during laser eye surgery, or we can add a contact lens there which changes the shape of the cornea to change the refraction of light, we can adjust someone's vision. We've got the pupil, which we should all know, that's the hole where the light goes through and then hits the lens. The lens is this object here that's kind of like a ball, and it's got liquid inside and it can change shape again to change the refraction of light. The iris is the coloured part of your eye, and those muscles can contract and relax to change the size of the pupil to adjust the amount of light that goes into the pupil. Now, I talked about the lens already. At the top and bottom of the lens, we've got the ciliary muscles and the suspensory ligaments, and the muscles can contract and relax to look at near and distant objects. We'll talk about that shortly. And also the ligaments can loosen or tighten. Images end up on the retina here, and we'll talk about the focal distance and the focal point, and really that focal point should land on the retina for a clear image. That image will land on the retina upside down, and then information gets converted into electrical impulses and removed to the brain via the optic nerve. Let's have a look at how we look at close and distant objects. And the reason I'm saying a close object, not a near object, is because close is a C, and in that situation the ciliary muscles contract. So we've got close, ciliary muscles contract, they're all the C's. Now in that contraction, this lens here becomes more C-shaped. It's kind of squashed in this way and wider this way. That gives a greater refraction of light, so it's become more C-shaped. So we've got a close object, ciliary muscles contract, allowing the lens to become more C-shaped. The suspensory ligaments are found at the top and bottom of that lens, and they loosen. They've got to loosen, because if you compare it to this picture over here, they're held tight in this picture, and it elongates it, it drags out that lens. So for the lens to become shorter and fatter, we must allow the suspensory ligaments to loosen. Like I said, makes the lens thicker and we get greater refraction of light. Over here then, for our distant objects, it's the opposite. So we've got the ciliary muscles are relaxing this time. Suspensory ligaments at the top and bottom are pulled tight. Becomes much thinner, refracts the light less. Adaptation to dim light. Now, if we go for bright light first of all, the radial muscles relax and circular muscles contract. Bright light, it's like we're able to function better, so we get our alliteration, okay? The radial relax and the circular contract in bright light. In dim light, it's like we've lost our way, we've got all confused and we lose our alliteration. So in dim light, radial contract, circular relax. Now, we saw a question recently and it just wanted contract as the answer, okay? But if you talk about radial and circular muscles, you would be just fine. Now let's move on to long and short-sightedness. So sometimes this normal eye can change shape and it becomes what we call rugby ball shaped, okay? So you can see this has elongated this way and kind of squashed. This one's elongated this way and kind of squashed this way. That affects where the image forms, okay? So where that focal length is will either be too short or too far and not land on the retina, and this causes short-sighted and long-sightedness. Let's have a look at how we can correct that. So short-sighted, there is a short focal length. You can see here, this focal point here should be landing on the retina, and it's landing much earlier. It's a short focal length. Now go with me on this. If I was in a cave and I was short-sighted, I'd be able to see everything in that cave. So short-sighted, we correct with a concave lens. What that concave lens does is it refracts out the light, you can see here, so that when it refracts in again by the lens, eventually we extend that focal length so it lands on the retina. Long-sighted, we're now looking at the opposites. We can see we've got a long focal length, okay? The focal point is actually beyond the retina. So again, we won't be able to see that correctly. So what can we do? Well, it's the opposite to the concave, so it's now the convex lens. What this does is it brings the beams in. So this one took the beams out. This brings the beams in a bit to reduce that focal length. Correcting defects. Well, I've talked about this already. I talked about the glasses just then. We've got the hard and soft contact lenses, which change the refraction of light at the cornea. We've got laser surgery to change the shape of the cornea. I mentioned that already. And we could replace the whole lens in the eye. Okay, temperature control. So this is controlled by the hypothalamus, which is the thermoregulatory centre. We've got, again, the use of alliteration to help us remember this, which is really handy. We can see here, these are blood vessels through the kind of extremities, so hands, feet, nose, ears, that sort of thing. And we can see here that this is in the cold conditions. There are muscles here either side of the capillaries, and when we're cold, those muscles contract, which constrict the blood vessels and therefore reduce the heat loss from the blood. So look at our alliteration. We've got cold, the muscles contract, which constrict the capillaries, reducing the blood flow. If we have a really extreme situation, someone has got a very cold, maybe they're in kind of Arctic conditions, something like that, and they're starting to get their frostbite, that is where the blood is completely shunted, redirected using this shunt vessel round here. So no blood is going through here. Well, if you've got no blood going through, you've got no oxygen going through, the cells die, and that's our frostbite. But that's in an extreme situation. The opposite of that then, in hot conditions, these muscles either side of the capillary there, they relax, okay? Muscles can contract or they can relax. When they relax, those capillaries dilate. That means they get bigger, so more blood flows through and there's more heat loss from those capillaries. And that heat loss you can see here because there's heat in the blood. As it gets close to the surface of the skin, that heat is lost from the body. Okay, let's have a look at all of the methods of controlling body temperature, and this goes back to homeostasis. Okay, so homeostasis was about maintaining body conditions, and this is maintaining body temperature. So when we're too cold, we've got that vasoconstriction. We just talked about that. Okay, blood vessels are constricted because the muscles contract, and that reduces the blood flow through those vessels. Muscles can also contract rapidly, causing us to shiver. That releases some heat energy, which acts as kind of like a blanket over our skin. And we've also got the hair muscles. We see our hairs stand on end when we're cold on our arms, and that's because the muscles, hair muscles, have contracted so that heat can be trapped in those hairs. If we're too hot, sweat glands release sweat, and when that evaporates from our skin, the heat is removed, leaving us feeling cooler. We notice this one a lot when we get out of a swimming pool. Okay, it's not sweat evaporating, but it's the water that's on our skin, and when we feel that evaporate, we do feel much colder. Okay, so sweating does exactly the same job. We've also got our vasodilation, which we just talked about. The muscles this time are going to relax, allowing more blood to flow because the blood vessels are wider, therefore more heat loss. And then lastly, the muscle hairs relax, our hairs lie flat so that heat can be lost more efficiently. Okay, moving on to the role of the kidneys. Let's look at our just initial information about what we know about water loss, urea and ions. Okay, so this is straight from the course content. Water leaves the body via the lungs during exhalation. If we breathe onto our hand, we can feel that moisture. Water, ions and urea are lost from the skin in sweat, but there's no control over how much is lost in the lungs or through the skin. Excess water, ions and urea are removed via the kidneys in the urine, and that's what we're going to talk about. And it's really important that cells don't lose or gain too much water by osmosis, because then they can't function efficiently. So that goes back to the importance of homeostasis. Let's look about where that urea comes from. And to do that, we need to go back to B2 in paper 1, where we learnt that we take in those proteins, we digest them into amino acids, because then they're small and soluble and can get into the bloodstream. And then some of those amino acids aren't used, okay? They're not used in protein synthesis, they're just wasted. So those amino acids, they head off to the liver. In the liver, the amino acids are deaminated into ammonia. Now, ammonia is really toxic, so really quickly, the liver then converts that ammonia into urea. So the liver creates urea from those wasted or unused amino acids. Now, we've got two directions that the exam board can go with the role of the kidneys. One is, how do we get rid of urea from the blood? And the other is, how do we control water loss from the blood through the kidneys? Okay, so this one is looking at the loss of urea. The next one is looking at that water control. Okay, so when you're looking at the question, don't just think kidneys, I know where I'm going. Think kidneys, right, are they asking about urea? In which case they may be asking about the loss of nitrogen, because there's nitrogen in the urea, because there was nitrogen in the amino acids, there was nitrogen in the ammonia, and therefore nitrogen in the urea. Or they may be asking about water. So be clear which one we're going for. So we can see here, we've got this kidney tubule, and there are thousands of these in the kidney. And then we've got a capillary, a blood capillary, coming really close to that tubule. Now, when this happens, the first process, there's three processes. The first one is filtration. Anything that is small and soluble is filtered from the blood into the tubules. Okay, so all of the water's coming across, all of the ions are coming across, all the urea and all of the sugar. Anything that is large, like large protein molecules, stay in the capillary throughout. Okay, so it's the water, the ions, the urea and the sugar, because they are small and soluble, means that they come into that tubule. Now, next thing that happens, selective reabsorption. We do not want any urea to go back into the blood. So all of the urea ends up down here. We'll talk about that in a second. All of the sugar is selectively reabsorbed back into the blood. Now, because it's all of it, has to happen by active transport. The amount of ions that is the correct balance that we need and the correct balance of water that we need is also selectively reabsorbed back. Water by osmosis, obviously, and ions by active transport. So that means that we've got going into the blood that's leaving the kidneys, we've got most of the water, most of the ions, and all of the sugar. So what's left? Okay, well, we've got all of the urea, some of the water and some of the ions going to the bladder in the form of urine. Okay, so the third stage is formation of urine. Let's have a look at the control of water. This is controlled by ADH. Now, ADH is letters, so we know it comes from the pituitary gland, anti-diuretic hormone. But if we just remember ADH, that's absolutely fine. So we've got our situation where we are quite dehydrated. There's not enough water in the blood, so we want to hold on to as much of it as possible. We don't want it to be lost in the urine. We want to selectively reabsorb back even more water. So when we're dehydrated, we go for more. Okay, more ADH is released. It makes the kidney tubules more permeable. Therefore, more water is reabsorbed back into the blood. So dehydrated, more ADH, tubules are more permeable, more water is reabsorbed back. Okay, and this is one of those negative feedback responses, and we'll look at those with the negative feedback loops in a bit. So if you're overhydrated, you've drunk lots and lots of fluids and you don't really need all of it, it's less. So less ADH is released, the tubules become less permeable, therefore less water is reabsorbed. Okay, so that's when we've had lots and lots of fluids, we don't need to keep hold of them all. We now go on to look at a classic evaluate question that will compare kidney transplants and kidney dialysis. This could be one of those questions where they give you lots of information and it's about making those comparison statements or using their subheadings, advantages and disadvantages for each of them to answer that evaluate question. Sorry about that. Okay, so if someone is unable to have a new kidney because they're on the donor list, they will be having kidney dialysis. And this is available to everybody who needs a new kidney, which is good news. And there's no need for immunosuppressant drugs. I'll explain about those when I talk about the transplants. Patients have to go and have this dialysis very regularly. So if you wanted to go on a long holiday, you wouldn't be able to because you'd have to have your dialysis. So it has a massive impact on a person's life. Overall, it's more expensive for the NHS because you're going to the hospital on a weekly basis, sitting there for a long time having your dialysis. So you've got a nurse with you, you've got the equipment, you've got the room. All of these things cost money. So overall, it is much more expensive for the NHS. And people who have dialysis, because it's all about removing those excess amino acids which come from our protein, they've got to be very careful with their protein intake to make sure they're not having these excess amino acids. So if we can reduce the amount of urea produced, then we've got a greater chance that we can kind of drag out the time between those sessions of dialysis. Now, someone on a kidney transplant, okay, it overall is cheaper for the NHS because although the actual operation is more expensive, it's a one-time thing, okay, and then it will last for quite a number of years. You don't have to watch your lifestyle as much, what you're eating and drinking, so that's quite handy. You get to live a more normal life. But the disadvantage is we have these immunosuppressant drugs. Now, we've talked about these before. Back in B3, when the pathogens come in our body, we said they've got these markers on them. They're called antigens. And all of these donor organs have these antigens on them that are kind of like flags saying, I'm not part of your body. And what the white blood cells do then is they cause an immune response. They try and fight that organ, which we would call rejection of the organ. To try and stop that happening, we take the immunosuppressant drugs. They do exactly that. They suppress the immune system. You will get ill more easily, but it's to try and stop that rejection of the organ. Obviously, we've got the disadvantage of there aren't many organ donors, and unfortunately that kidney will not last forever, about 8 to 9 years. And obviously with any operation there is a risk. So that's going to help us with our evaluate question comparing the two. Let's have a look at how dialysis works. So it's got this stuff in it called viscing tubing. It's a partially permeable membrane. It allows those small soluble molecules to move out of the viscing tubing or indeed into the viscing tubing. Here we've got the blood coming from the patient, probably from their arm. Okay, it's coming through here and it's going this way, wiggling through the dialysis machine because it gives it a larger surface area. It gives it more opportunities for that diffusion of urea to come out. So all of the urea moves from this tubule, this viscing tubing, into the dialysis fluid by diffusion. Now, we talked about countercurrent in fish in B1, and we've got a countercurrent system here as well. That means that the blood is moving this way and the dialysis fluid is moving this way, and that means that we get diffusion at all points. Okay, so the dialysis fluid goes into the machine. It's heated up. It's heated up so that we don't lose our body temperature, okay, to maintain that homeostasis of the body temperature. We heat the dialysis fluid. Okay, because all of that heat would be lost from the blood, and then when the blood goes back into the person, it would be a lot cooler, and we don't want that to happen. So the dialysis fluid is very fresh here. Here, the dialysis fluid has no urea in it. But if we think about it, there's been urea diffusing out of these viscing tubing all the way along. So the amount of urea in this part of the blood is very, very low. But because we've got no urea here, we get this diffusion across. At this end, this dialysis fluid is just about to go into the used container, so it's very high in urea. But the urea in the blood is even higher because it's just entering the machine. So again, urea can diffuse across. At every point in this machine, there is a diffusion gradient, so urea can leave that viscing tubing and get into the dialysis fluid. Let's have a look at some of our negative feedback loops, okay? So these are things we've already talked about, temperature control. If someone is too cold, then the muscles contract, which constricts the blood vessels and less blood passes through the vessels, less heat loss. If someone is too hot, then those muscles relax, blood vessels dilate and more blood passes through the vessels. If we've got the water control, so this was the kidneys with the ADH, if there is too much water in the blood and we don't need all of it, then it's less. Less ADH is released from the pituitary gland, makes the tubules less permeable, and therefore less water is selectively reabsorbed. Whereas if we've got too little water in the blood, we need to pull back as much as possible. We've got more, so more ADH, therefore tubules are more permeable, more water is selectively reabsorbed. And you can see on both of those I was kind of creating little 3-mark answers. So 3 marks about being cold, 3 marks about being hot, that gives us 6 marks. And the same on the ADH. Our last section then, we're looking at hormones in plants. As we know, they keep adding these bits of plants on the back of the topics. And this one is, so the hormone is auxin, okay, plant hormone auxin. And auxin, if we just remember this, auxin in shoots speeds up growth, auxin in roots slows down growth. So we can see here that the auxin's in the shoot and it can redistribute itself based on a response to a stimulus. And the stimulus here is light. So the light is shining from this side. The auxin has redistributed itself to build up on this side, the shaded side of the shoot. That makes those cells grow faster, so it bends towards the light, which then helps photosynthesis. Okay, so auxin in shoots speeds up the growth on the shaded side. Now we've got a slightly more difficult example, but this is the one the exam board love. Okay, this is geotrophism. They can't decide whether they want to call it geotrophism or gravitrophism, so they go with both. Choose your favourite. And this is where the plant has fallen over. Okay, usually they say it's in a dark place. If they talk about it being dark, you've got to talk about this one. So let's have a look. The auxin goes to the underside of the shoot and the underside of the root. On the underside of the shoot, speeding up the growth, allowing it to go away from gravity. Don't say to where the light might be. Say it allows it to grow away from gravity. It's a negative trophism away from gravity. So the auxin builds up here. Auxin in shoots speeds up growth, so it grows faster round this side up away from gravity. Auxin in roots slows down growth, so the auxins build up on this side, on the underside again, so they grow slower, so the other ones come down more, it comes down more here, allowing that root to bed back into the soil. So auxin in shoots speed up growth, auxin in roots slow down growth, and it's both the auxin going to the underside. Uses of plant hormones, the final slide. So auxin, we can use it in rooting powders. So if we take a cutting from a plant, we dip it in some rooting powder, some auxin, and then we can plant it into the soil, help it grow. Gibberellins, just remember gibberellins and germination, so it helps seed germination, fruits and flowers. Quite often these are match-up ones, so just kind of gibberellins, germination should be enough to help us. And then when we pick the fruit from the tree, it's not ripe, okay, mainly because we get a lot of our fruit from far away, so we don't want it to be rotten by the time it arrives. So we pick it when it's not ripe, and then ethane is sprayed on it and it ripens on transit. So when it's, by the time it gets to the supermarket, it's the way we want it to be. Okay, hope you found that useful. Thank you.